The fallopian tubes, also known as uterine tubes or salpinges, singular salpinx, are tubes that stretch from the uterus to the ovaries and are part of the female reproductive system. The fertilized egg passes through the fallopian tubes from the ovaries of female mammals to the uterus. The fallopian tubes is simple columnar epithelium with hair-like extensions called cilia which carry the fertilized egg. In other animals, the equivalent of a fallopian tube is an oviduct. The name comes from the Catholic priest and anatomist Gabriele Falloppio, for whom other anatomical structures are also named. The fallopian tube is composed of 4 parts. These are, described from near the ovaries to inwards near the uterus, the infundibulum with its associated fimbriae near the ovary, the ampulla that represents the major portion of the lateral tube, the isthmus, which is the narrower part of the tube that links to the uterus, and the interstitial, or intramural, part, the narrowest part of the uterine tube, that crosses the muscles of the uterine. The average length of a fallopian tube is 11 to 12 centimeters. The uterus opens into the fallopian tube at the proximal tubal opening, also called the proximal ostium or OS, after the uterotubal junction, and accessible via hysteroscopy. Occlusion at this opening is referred to as proximal tubal occlusion. From there are 3 named parts of the fallopian tube: the isthmus, the ampulla, and the infundibulum. The isthmus sits next to the opening of the fallopian tube into the uterus. It connects to the ampulla, Latin, flask, which curves over the ovary and is the most common site of human fertilization. The ampulla connects with the infundibulum, which rests above the ovaries, and ends at the distal tubal opening, or abdominal ostium, into the abdominal cavity where, in ovulation, the oocyte enters the fallopian tube. The opening is surrounded by fimbriae, which help in the collection of the oocyte. Occlusion of this opening is referred to as distal tubal occlusion. The fimbriae, singular fimbria, is a fringe of tissue around the ostium of the fallopian tube, in the direction of the ovary. Of all fimbriae, 1 fimbria is long enough to reach the ovary. It is called fimbria ovarica. An ovary is not directly connected to its adjacent fallopian tube. When ovulation is about to occur, the sex hormones activate the fimbriae, causing them to swell with blood and hit the ovary in a gentle, sweeping motion. An oocyte is released from the ovary into the peritoneal cavity and the cilia of the fimbriae sweep the ovum into the fallopian tube. When viewed under the microscope, the fallopian tube has 4 to 5 layers, depending on the classification system used. From outer to inner these are the serosa, subserosa, muscularis, submucosa, and innermost mucosa with lamina propria and epithelium. The serosa is derived from the visceral peritoneum. The subserosa is composed of loose adventitious tissue, blood vessels, lymphatics. The muscularis consist of outer longitudinal and intercircular smooth muscle coats. This layer is responsible for the rhythmic contraction, called peristalsis, of the fallopian tubes. The histological features of tube vary along its length. The mucosa of the ampulla contains an extensive array of complex folds, whereas the relatively narrow isthmus has a thick muscular coat and simple mucosal folds. The innermost layer of the tube is an epithelium composed of a single layer of column-shaped cells. The columnar cells have microscopic hair-like filaments called cilia throughout the tube, most numerous in the infundibulum and ampulla. Estrogen increases the formation of cilia on these cells. Between the ciliated cells are pegged cells, which contain apical granules and produce tubular fluid. This fluid contains nutrients for spermatozoa, oocytes, and zygotes. The secretions also promote capacitation of the sperm by removing glycoproteins and other molecules from the plasma membrane of the sperm. Progesterone increases the number of PEG cells, while estrogen increases their height and secretory activity. Fluid flows through the tubes towards the ovaries, the opposite direction to the action of the cilia. Embryos develop a urogenital ridge that forms at their tail end and eventually forms the basis for the urinary system and reproductive tracts. Either side into the front of this tract, around the 6th week develops a duct called the paramesonephric duct, also called the Mullerian duct. A second duct, the mesonephric duct, develops adjacent to this. Both ducts become longer over the next 2 weeks, and the paramesonephric ducts around the 8th week cross to meet in the midline and fuse. One duct then regresses, with this depending on whether the embryo is genetically female or male. In females, the paramesonephric duct remains, and eventually forms the female reproductive tract. The portions of the paramesonephric duct which are more cranial, that is, further from the tail end up forming the fallopian tubes. In males, because of the presence of the Y-sex chromosome, anti-Müllerian hormone is produced. This leads to the degeneration of the paramezonephrine duct. As the uterus develops, the part of the fallopian tubes closer to the uterus, the ampulla, become larger. Extensions from the fallopian tubes, the fimbriae, develop over time. Apart from the presence of sex chromosomes, specific genes associated with the development of the fallopian tubes include the WNT and HOX groups of genes, LIM1, PAX2, and EMX2. Embryos have two pairs of ducts to let gametes out of the body. One pair, the Müllerian ducts, develops in females into the fallopian tubes, uterus, and vagina, while the other pair, the Wolffian ducts, develops in males into the epididymis and vas deferens. The homologous organ in the male is the rudimentary appendix testis. The fallopian tube allows passage of an egg from the ovary to the uterus. When an oocyte is developing in an ovary, it is surrounded by a spherical collection of cells known as an ovarian follicle. Just before ovulation, the primary oocyte completes meiosis to form the first polar body in a secondary oocyte which is arrested in metaphase of meiosis II. At the time of ovulation in the menstrual cycle, the secondary oocyte is released from the ovary. The follicle and the ovaries wall rupture, allowing the secondary oocyte to escape. The secondary oocyte is caught by the fimbriated end of the fallopian tube and travels to the ampulla. Here, the egg is able to become fertilized with sperm. The ampulla is typically where the sperm are met and fertilization occurs. Meiosis II is promptly completed. After fertilization, the ovum is now called the zygote and travels towards the uterus with the aid of the hair-like cilia and the activity of the muscle of the fallopian tube. The early embryo requires critical development in the fallopian tube. After about 5 days, the new embryo enters the uterine cavity and on about the 6th day implants on the wall of the uterus. The release of an oocyte does not alternate between the two ovaries and seems to be random. After removal of an ovary, the remaining one produces an egg every month. Salpingitis is inflammation of the fallopian tubes and may be found alone, or as part of pelvic inflammatory disease, PID. A thickening of the fallopian tube at its narrow portion, due to inflammation, is known as salpingitis myconodosa. Like PID and endometriosis, it may lead to fallopian tube obstruction. Fallopian tube obstruction may be a cause of infertility or ectopic pregnancy. Fallopian tube cancer, which typically arises from the epithelial lining of the fallopian tube, has historically been considered to be a very rare malignancy. Recent evidence suggests it probably represents a significant portion of what has been classified as ovarian cancer in the past. While tubal cancers may be misdiagnosed as ovarian cancer, it is of little consequence as the treatment of both ovarian and fallopian tube cancer is similar. Occasionally, the embryo implants into the fallopian tube instead of the uterus, creating an ectopic pregnancy, commonly known as a tubal pregnancy. While a full testing of tubal functions in patients with infertility is not possible, testing of whether the tubes are open, called patency, is important as tubal obstruction is a major cause of infertility. A hysterosalpingogram, laparoscopy, and dye, or hysterocontrast sonography will demonstrate whether the tubes are open. Tubal insufflation is a standard procedure for testing patency. During surgery, the condition of the tubes may be inspected and a dye such as methylene blue can be injected into the uterus and shown to pass through the tubes when the cervix is occluded. As tubal disease is often related to chlamydia infection, testing for chlamydia antibodies has become a cost-effective screening device for tubal pathology. The surgical removal of a fallopian tube is called a salpingectomy. To remove both tubes is a bilateral salpingectomy. An operation that combines the removal of a fallopian tube with removal of at least one ovary is a salpingo-oophorectomy. An operation to remove a fallopian tube obstruction is called a tuboplasty. The fallopian tube can prolapse into the vagina and can be mistaken for a tumor. When this happens, it is usually after a hysterectomy. The fallopian tubes are named after the 16th century Italian anatomist Gabriele Falloppio, the first person to provide a detailed description of the tubes. He thought they resembled tubas, the plural of tuba in Italian being tube, which was misunderstood and became the English tube. Though the name fallopian tube is eponymous, it is often spelt with a lowercase f from the assumption that the adjective fallopian has been absorbed into modern English as the de facto name for the structure. Kerman, Robert J. Ellenson, Laura Hedrick, Ronit, Bridget M., eds. 2011. Diseases of the fallopian tube and paratubal region. Blaustein's Pathology of the Female Genital Tract, 6th ed. Springer. ISBN 9781441904881.